Welcome, everybody. My name is Sarah Booth, and I'm here to introduce today's speaker for the Monday seminar series at the HNRCA. Our speaker today is Dr. Daniel Promislow, who has worked for over 30 years on the biology of aging. Daniel joined the HNRCA and Tufts last month, and he brings to Tufts his expertise in quantitative genetics, biodemography, and comparative systems and theoretical biology and aging research. After graduating from the University of Chicago, Dr. Promislow won a Rhodes Scholarship to attend Oxford University, where he earned his doctoral degree in 1990. During his graduate work, he began studying aging, publishing the first definitive comparative evidence that aging is widespread in natural populations, which at the time was contrary to popular belief. He then carried out his postdoctoral work at Queen's University in Canada and at the University of Minnesota. It was during that time that he began studying the genetics of aging in the fruit fly, a widely used model system in aging. Dr. Promislow's work centers on the problem of understanding the genetic basis of aging in natural, genetically variable populations, trying to determine whether mechanistic pathways that affect aging in lab-adapted, inbred organisms can account for the variation rates of aging that we see in natural populations, including humans. Dr. Promislow has received many awards for his work and is a fellow of both the American Aging Association and the American Association for the Advancement of Science. Dr. Promislow is committed to mentoring and indeed co-authored the book, The Chicago Guide to Landing a Job in Academic Biology. Dr. Promislow is also the principal investigator of the NIH-funded Dog Aging Project, which has recruited more than 50,000 dogs across the United States. His seminar today will describe the tremendous potential this project has to create a better understanding of the causes and consequences of aging, not only in companion dogs, but also in humans, given that dogs suffered from a very large number of the same diseases as we do. They live in our environment, and the medical care for our companion dogs is second only to humans. I encourage the audience to enter your questions in the Q&A section during the course of the seminar. And with that, I'm going to hand it over to our speaker, Dr. Daniel Promislow. Take it away, Daniel. Thank you.
Thank you so much, Sarah, for your really kind introduction. It's unusual for me to be able to say that I'm excited to be here in both senses of the word. Here as a visiting speaker and also here as a new senior scientist at the HNRCA. I am really excited to begin interacting with my colleagues here at HNRCA and at Tufts and also across the Boston area. I think the potential for new and exciting work in aging is just phenomenal. I just want to start with 3 disclosures. I'm on the scientific advisory boards of Invoxia and Wonder Health, and I'm chair of the board and treasurer of the nonprofit Dog Aging Institute. So when I was starting out in the early 1990s in aging as an evolutionary biologist, around the same time, some really exciting discoveries were happening in the molecular genetics of aging. Tom Johnson really set the stage with his discovery of age1. But at the time, we didn't know what the gene was. And then Cynthia Kenyon, whose work is shown on the top, showed that when you knock out this gene DAF2 in worms, you can see the survivorship as a function of age in days goes from a mean of about 20 days to a mean of more than 40 days. So knocking this gene out more than doubles the lifespan of a worm. Gary Ruvkin, who's here at MGH in Boston, showed that this was a Dower gene, and many genes in the Dower pathway have been shown to affect lifespan. And we now know more generally that in these laboratory organisms that Sarah referred to in her introduction, in worms, in fruit flies, and in laboratory mice, knocking out genes in the insulin-like signaling pathway typically extends lifespan. And this raises the big question of whether what we see in these highly inbred isogenic lines, where the only thing that differs between short and long-lived lines is one gene, and we control for all the environmental factors, can that translate to the real world? And this famous photograph from 1914 from Connecticut State Agricultural College shows cadets lined up from shortest to tallest, forming this nice Gaussian distribution. This was all men at the time. There are similar photographs now, including women, and they show the same Gaussian distribution, just shifted to the left. All of this variation is the phenotypic variation. And the source of all of that variation is entirely due to two factors: genetic variation and environmental variation. Of course, within each, we can tease them apart, and there's lots of different types of genetic and environmental variation. But there's a fundamental equation, the only equation I'm going to show today, in quantitative genetics, which tells us that the phenotypic variation in a population like that which you see here for height is due entirely to environmental variation, genetic variation, and the covariance between the two. That is, how does the effect of a gene depend on the environment in which it happens? Or how does an environmental factor influence the phenotype dependent on the genetic background? And I stress this because this is really what I want to spend some time talking about today. In thinking about precision medicine, I'm going to talk about the Dog Aging Project, but I also really want to encourage us to begin asking ourselves, what does it mean to do precision medicine? How do we do it? How can we do it? What are the challenges in human populations? Another way of illustrating that equation is with this figure, that genes and environment ultimately shape downstream phenotypes, in this case, aging. And often when we talk about precision medicine or precision science, precision geroscience in the case of aging, we think about a framework where the mechanisms that are intermediate between upstream genes and environment and downstream phenotypes like aging are what we study to do precision geroscience in this case. And the mechanisms tell us how it is that genetic or environmental factors shape aging. So that's generally the framework that I think about, and I think many people think about. Sometimes we talk about the networks that we can analyze that help us understand genetic factors, the molecular mechanisms, and downstream phenotypes. But and just to flesh it out a little bit more, we can think about all of the different domains, the omic domains, the genome, epigenome, transcriptome, proteome, metabolome, microbiome, and so on, that ultimately impinge on phenotypes of interest. The environment is here. I've drawn this arrow going backwards as well because, for example, there are metabolites that can influence the epigenome. When you think about acetylation, for example, or methylation. So this is one way that we can think about precision medicine, and a way that we often do. But I want to suggest a second framework, and I think its value is both in suggesting a way to think about precision science, precision geroscience, precision nutrition, but also raises some challenges and maybe some approaches we can use to think about it. And it's really this framework. And I was at a workshop last week on sex and gender and precision aging and precision medicine. And as I was thinking about my presentation, I used this framework, and it made me realize that we have some challenges, but also that model organisms might help us address those challenges. So what's the second framework? So here we are. Let's just imagine that we have measured some trait. Think of your favorite trait that you could measure in humans. And there is a measure before treatment and after treatment. Let's say that the treatment is a dietary intervention. The dashed line is the isometric line where x equals y. And you can see that after treatment, individuals tend to have a greater value. So they tend to be above the line. And so that's sort of the main effect, but there's a lot of variation too. And so if we look at the residual variation around the isometric line, how much have individuals changed since the treatment? There's all this variation. Is the signal or is this noise? That's really our challenge. And in human studies, we are all unique. We have a unique genome, other than if we're an identical twin, and we have a unique environmental experience, both intrinsic environmental experience, hormonally, for example, and extrinsically for our whole lives, starting from conception. So how do we tease apart what's signal and what's noise? And that's where I'd like to suggest that these model organisms actually can help us think about it. So now imagine that instead of looking at humans, we're looking at fruit flies. And each one of these points represents the average of tens or hundreds or even thousands of individuals within a single co-isogenic population, isogenic population. So this is not noise. This is highly reproducible variation. So this simple framework captures the genetic variation. There are, let me just change my pointer here. Here we are. So there are high value genotypes and low value genotypes. So this axis is genetic variation. There's the main effect. So this treatment clearly makes individuals have a higher value of whatever the trait is that we're measuring, although there's a lot of variation. And now we can say that all this variation is actually, in this case, gene by environment interaction, that the degree of the effect, there is a main effect, and that's the E, the treatment, the environment. Say we've altered levels of some metabolite, but the degree to which each genotype changes is a gene by environment interaction. And so that's actually what we can measure. And that I want to suggest to this audience is actually the way for us to think about precision science, precision medicine, precision nutrition, precision geroscience, is to understand more deeply these interactions within the context of this equation, that it's this covariance that's the interesting stuff from a precision perspective. So if we're looking at, let's say, methionine restriction, there is a main effect, but more of the variation is actually due to the covariance. So the methionine restriction impacts both the environmental variance and the covariance. So just a couple of examples from fruit flies, and then I'm going to shift into dogs. So here we're looking at a set of 140 genotypes from the Drosophila genetic reference panel. This is a set of inbred fly strains, much like the inbred mouse strains that we see up at Jackson Labs. And so you can see for lifespan, there are long-lived strains and short-lived strains. In this particular population, on average, males live a little bit longer than females in this particular environment. And there's a lot of this residual variance, the genotype by sex interaction. So how much longer males live relative to females is actually highly dependent on genetic background. We can use the same kind of framework to think about an intervention. So here, for example, I'm showing you how treatment with rapamycin across these same lines delays development. So in some lines, there's no response to rapamycin. In others, development is delayed by almost 100%. Typical development from egg to pupa is about 8 days. And you can see that the delay is up to about 7 days in treated genotypes. And I'll just mention in these resistant lines, we've tried experiments where we've increased rapamycin 500-fold, and they still don't respond. We're just writing up that work now. Treated lines tend to be smaller, and what I'm showing you on the right here are the control lines with the ethanol vehicle and the treated lines with rapamycin. And females are shown in red. They're much larger than males, and fruit flies shown in blue. On average, the treated lines are a little bit smaller, but you can see the lines are crossing over. There's a lot of interaction. So if we put all of that together into a model, I just want to show you here, there's a significant effect of treatment. Treatment makes flies smaller. A significant effect of sex, a huge effect. We already know that. Males are smaller than females. But I want to show you this highly significant treatment by sex by strain effect. What that means is, in general, rapamycin makes flies smaller, but the degree to which it does so is sex specific depending on the strain. In some strains, it has a bigger effect in males. In some strains, it has a bigger effect in females. So this is a powerful framework to begin to tease apart these precision effects. So we're not there yet with dogs. We're just beginning, but that's really the framework that I have in mind as I'm thinking about some of the initial studies that I'm going to tell you about today, much of which we're just writing up now. So I'm going to share quite a bit of unpublished work. So the Dog Aging Project is a nationwide long-term longitudinal study whose primary goals are to identify the genetic and environmental determinants of healthy aging in companion dogs. This is our previous family dog, Frisbee. In her healthier years, she's no longer with us, but she lived to 16 and a half, which for a 45-pound dog is an extremely long lifespan. But here she is on top. That's Mount Rainier in the background. She was a great hiker. And then in her later years, she really struggled with musculoskeletal challenges, probably sarcopenia, certainly osteoarthritis, as well as pancreas problems, and I'll come back to that. Why dogs? This actually was really stimulated by a paper that came out. I will tell you another time that in my life, my scientific path has been changed 3 times by papers. So that's one of the reasons I tell students in my lab to read science widely, scientific papers. When this paper came out from Elaine Ostrander's group, I got really excited. I wasn't working on dogs at the time. This paper was all about the genetics of size and found that a single locus, the insulin growth factor 1 locus, explains about half the variation across breeds that large breed dogs like this Great Dane tend to be fixed for the ancestral IGF-1 allele that's found in wolves, and toy breeds like this little Chihuahua tend to be fixed for a novel IGF-1 allele that arose thousands of years ago. Interestingly, IGF-1, when knocked out, as I alluded to at the top of the seminar, reduces size and increases lifespan in mice. And so I immediately got excited about whether this might have something to do with aging in dogs. In my graduate work, as Sarah alluded to, I focused on aging in natural populations. And this is not my figure. This was from a paper that came out many years later. But we've known, in fact, going back to Aristotle and Francis Bacon, we've known that large species tend to be long-lived. So here we've just got, in both birds and mammals, larger species, longer-lived. Interestingly, in dogs, it's the opposite. The large breeds are shorter-lived. So when this paper came out, I immediately wanted to know whether the reason that large breed dogs are short-lived is because of this allelic variation in IGF-1. We still don't know the answer, but it got me addicted to studying aging in dogs and creating this big team science project. So I'm thankful, permanently thankful to Elaine Ostrander for the inspiration, and she's been a great supporter all along the way. So why else dogs? Any of you who have a dog or go to a dog park will see tremendous morphological variation, most of which is genetic. All of these dogs are descended from a single species. These are all considered common but separate species from the ancestral wolf. Tremendous environmental variation. There are rich dogs. This is a dog in front of its doghouse. And not so rich dogs. There are healthy dogs that like to hike and not so healthy dogs that, at least this one, smokes. These are pre-COVID numbers. There are certainly now well over 100 million dogs in the first year of COVID. Millions of people adopted dogs. Tremendous behavioral variation. Herding dogs and jumping dogs and digging dogs and swimming dogs, and even at least one cycling dog, lots of behavioral variation. And importantly for us and for this audience, I think will be of interest that within each purebred breed, certainly, and these are recapitulated in the mixed breed dogs, there are different spectra of diseases. So I'm just showing you four main causes of mortality. So first in blue, respiratory diseases, most common cause of mortality in the bulldog. In red, heart disease, which is quite common in the small breed dogs. Notably, dogs do not get cardiovascular disease. They don't get heart attack or stroke, but they do get cardiomyopathy, mitral valve disease, and so on. Retrievers tend to get leukemia, lymphoma, and then this interesting breed, the Shar-Pei, gets stomach and intestinal diseases. The wrinkly coat in the Shar-Pei is caused by high levels of hyaluronan, and there are some in the aging field who believe that at least the naked mole rat is as extremely long-lived as it is because of its extremely high levels of high molecular weight hyaluronic acid. Whether that will protect the Shar-Pei from cancer, as some people like Vera Gorbanova believe it protects the naked mole rat, is an interesting question, remains to be seen. And finally, of course, people love dogs, and so they are motivated to become community scientists with us on our project. So we launched in 2019, recruited our first dogs in 2020. We hadn't planned for a massive global pandemic, but we managed to be pretty successful anyways. As Sarah mentioned, we've now recruited 50,000 dogs. And very briefly, the way it works is people go to our website. They find out about us through mainstream media, social media, word of mouth, and so on. Go to our website, dogagingproject.org, where they sign up their dog. Once they complete a survey using our RedCap portal, so it's highly secure, it's a 10-part survey. Once completed, they become a member of the pack. And within that pack, there are other cohorts that I will tell you about that require visits, in some cases, to the veterinarian. All of the data ultimately end up at our data center, which is here in Boston at the Broad Institute using their Terra platform. We have a biobank at Cornell Veterinary School of Medicine with currently about 15,000 samples in it. We do diagnostic labs and others that I'll tell you about. And then ultimately, we push the data back to the owners in the cases of genetic reports and to the scientific community through papers and so on. So we've been supported by U19 from the National Institute on Aging. And the general structure of the project recapitulates that picture that I showed you earlier. So genetic information, there's whole genome sequencing, as well as owner-reported breed information, which sometimes is more reliable than others, as you might imagine. Lots of environmental information, publicly available data on air quality and water quality and green space, environmental data reported by the owners. And then in some cases, we provide kits to the owners to collect water, to put silicone tags on the dogs to collect volatile organic compound information that we measure using GCMS, and so on. Phenotypes, morbidity and mortality, frailty instruments, and so on. And then that intermediate part, we do standard CBC Chem UA as you would do in humans, as well as the plasma metabolome, PBMC epigenome, and flow cytometry or facts on the PBMCs, fecal microbiome, and so on. All of these data go into several specific discrete projects that interact with each other. Project one is really just describing aging in dogs. Interestingly, while there are many different specialties in veterinary medicine, like orthopedics or ophthalmology or dermatology, there is no geriatrics. And so we're kind of laying the groundwork for that. Project two is based partly here in Boston at the Broad Institute and UMass with Eleanor Carlson, and then Josh Akey leads it at Princeton with Eleanor, looking at the genetic basis of all that variation that we measure in Project one. Project three is the systems biology with myself, Noah Snyder-Mackler in Arizona, Elfhanan Borenstein in Tel Aviv. I should mention Project one is led by Kate Creevy. Kate is the co-founder with me and Matt Kaberlein of the Dog Aging Project. I should have mentioned when that science paper came out in 2007, at the time I was at the University of Georgia, I reached out to the vet school because I didn't know anything about dogs. And everybody there was sort of playing hot potato. They didn't have time to talk to some guy in genetics about dog aging. So they put the newest faculty member onto me, Kate Creevy, who emailed and said, I don't know about aging, but I'm happy to talk to you. And of course, now she's the chief veterinary officer and the leader of Project One. There's a separate study that's kind of a spin-off of the dog aging project that is a clinical trial that will ultimately be in 580 dogs. So far, there are about 150 that have been recruited. We call it TRIAD, or Test of Rapamycin in Aging Dogs, testing the ability of low-dose rapamycin for 6 months to improve heart function, cognitive function, and longevity in dogs. So I mentioned that we have the Dog Aging Project PAC. So far, we've recruited just over 50,000 dogs. And then nested within that are other groups. There's Foundation. These are dogs that whose owners provide us with the veterinary electronic medical records and a cheek swab for DNA. And so we can do genetics of aging in these dogs. A separate cohort that we call Precision has everything that the blue cohort has, plus we send a kit that the owners bring to the veterinarian to collect hair, blood, fecal samples, urine, and that's what we do, the omics. So they have the MRs and the genetics plus all the omics. There's the intervention study. There's another spinoff that's a brain health study looking at dogs with what we call canine cognitive dysfunction, which looks very much like Alzheimer's disease in dogs. And we will be recruiting a cohort of extremely long-lived dogs for their size. We haven't done that yet. I mentioned all the biospecimens. I'll just skip through this. And then we measure all kinds of aging-related traits. So just 2 of hundreds. Not surprisingly, as any of you who have dogs will know, as dogs age, they become less active. And we see that here. This is owner-reported time spent outside, active, on leash. And you can see that dogs decline in activity with age. But interestingly, and maybe not surprisingly, there's greater activity in rural dogs, more time spent outside than in suburban and urban dogs. The numbers out above 15 are low. And because we recruited anyone who came forward, those who signed up very old dogs were biased towards healthy dogs because people will not sign up a dog that is in its last weeks or months for a study of healthy aging. So you see that uptick here, partly due to bias, partly due to small sample size. We see an effective age on cytokines here, looking at IL-2, IL-6, IL-8, and TNF-alpha, all of which increase with age. So we've measured all kinds of things, and we're just beginning to tie those measures to measures of disease. And this was actually our first genetic study. I'm showing you on the left a video of Frisbee in her later years. She's about 14 and a half here, and you can see this is not a dog who's able to hike up mountains anymore. Interestingly, the first question we asked that Eleanor Carlson's group asked is, can we identify any single nucleotide polymorphisms or SNPs that are associated with owner-reported mobility? We got 2 hits. This one here, SH3BP4, is a really interesting hit because it turns out to be a negative regulator of the target of rapamycin pathway, which is a great interest to aging. This was work done by Kathleen Morrill, who's a graduate student at the time, since graduated. We have not yet gone back to validate this on a larger cohort. We're just now starting to do that. This was on an initial cohort of about 2,000 dogs, so whether this holds up is an open question. But you can see visually that musculoskeletal challenges are a major issue for all dogs, especially mid-size and large breed dogs as they get older. So I also want to stress the importance of this being a longitudinal study. Those of you who work on human longitudinal studies appreciate that when we do cross-sectional studies, there are some serious challenges around correlation versus causation. And I just want to describe one study. This is a study done by a terrific postdoc at the time, Emily Bray, who is now on faculty at the University of Arizona. Using our initial studies, Emily asked whether there was a relationship between feeding frequency and the canine social and learned behavior score. This is a survey that was modified from what we call the CBARC survey, which is a measure of cognitive function in dogs. Higher scores are worse cognition. Above 50 suggests canine cognitive dysfunction. And while it was a small effect size, what you can see here is the black line is dogs that feed more than once a day, the dashed line, dogs that feed just once a day. And at least at older ages, those that feed once a day are slightly less likely to have canine cognitive dysfunction. It was a weak effect, but significant, partly because of this very large sample size, many thousands of dogs. So in a cross-sectional study, correlation between feeding frequency and the CSLB score. The motivation for doing this study is that in the lab, there's some suggestion that intermittent fasting increases health span, healthy aging. And so feeding once a day for dogs is kind of like intermittent fasting. It's a 23.5-hour fast for every dog. The way this was picked up in the media was that it was as though we were saying something causally. So here are a few headlines. How often should you feed your dog? Giant studies gives a surprising answer. Scientists surprised by how often you should actually feed your dog. Here's how often you should feed your dog. We did not say that. We just said, here's the correlation. I'd like to give an example of why this is a bad idea to draw conclusions about cause from correlation. So if you look at the letters in the winning word of the Scripps National Spelling Bee, you see an almost perfect correlation over several years with the number of people killed by venomous spiders in the United States. Obviously, these two are highly correlated, but there's no causal relationship, at least none that I can think of. So in the case of this particular relationship, I thought back to my own dog's history. So if you look at my dog Frisbee's veterinary EMR, here she is, golden retriever mix. This is actually not true. That's what the vet said. She was actually a Chow Shepherd Spaniel mix. So it's not surprising that owners often don't know what their dog is. Female spayed, 40 pounds. And here you can see extremely high levels of a biomarker of pancreatitis. And when she was diagnosed with pancreatitis, the veterinarian said, you need to do two things. Start feeding her many small meals throughout the day, and you're going to have to cook her own food. And here I am cooking Frisbee's food. And I caution you not to do this unless the veterinarian tells you to do this. I am not saying you should cook your dog's own food. But of course, unhealthy dogs are often going to be told to not feed once a day. So it might be that the causal relationship is actually going in the other direction. So let's get back to precision medicine in dogs. I want to talk a little bit about the kinds of mechanistic work that we're looking at, and especially that Project 3. And these are just some preliminary results. And in a way, what I'm really saying is that I think the path forward for precision geroscience is kind of coupling that first framework, looking at omics and networks, with the second framework, thinking about those interactions, those gene by environment interactions. So while breeds are not like fruit fly strains or mouse strains where we have tens or hundreds of individuals that are genetically identical, they do form genetic silos. So there's plenty of genetic variation within a golden retriever or a Chihuahua or a Shar-Pei, but the genetic variation between them is pretty considerable as well. And so in a way, we can think of the dog breeds as giving us similar measures within a breed of phenotypes based on relatively common genetic effects and environmental factors. I'm not going to show you that kind of analysis that we've been doing in the fruit fly, but I think this might be a way forward, and we can talk about statistically some of the challenges and how we might do that. So just to give you some early results from my collaborators, Noah Snyder-Mackler, who's been working on the epigenome, Elhanan Borenstein, and his team working on the microbiome, and my own group working on the metabolome. So there's no end to the kinds of questions we can ask. We're looking at geographic variation in dogs, definitely interested in diet. We have owner-reported information, including in many cases scans of the actual barcodes from the dog food that the owners are buying. So we can tell you quite a bit about the kind of food that they're eating. Lots of information about sex, and veterinarians talk about 4 sexes in dogs, male and female, but also intact and sterilized. And I'll mention that a little bit, activity levels and so on. So just a few quick results. So first of all, as many of you know, there are some pretty considerable changes as we age in the microbiome. And as in recent years, people have come to really appreciate the importance of the microbiome, both in terms of cause and consequence in relation to aging and age-related disease in human populations. And of course, as Rob Knight has shown some years ago, the dog microbiome and the human microbiome share quite a bit. Given a population of dogs and their owners, you can actually match each dog to their owner through the microbiome because we share an environment. And if you think about dogs licking us and trying to, licking our environment, and we touch our environment, there's lots of exchange there. In the dog, we see a decline in both alpha diversity. So this is a measure of the diversity of each sample, as well as an increase in beta diversity. So that's the divergence among individuals that we can think of this as the microbiome becoming less regulated in dogs as they age, so they sort of drift apart. And we have lots of information on the different kinds of microbes. This is a team based at Tel Aviv University, and they're working on how this variation relates to health and disease in dogs. There's a tremendous amount of interest in the aging field in developing age predictors. So what you're seeing here in the dog, and this is actually a pilot study that was carried out by a graduate student that was co-supervised by myself and Noah Snyder-Mackler. You can see that there's this pretty tight relationship between chronological age and predicted age, based in this case on methylation patterns across the entire genome. This was work done by Kelly Jin, and interestingly, she looked at the methylome. She also used a TAC-seq, which is a way to measure chromatin accessibility. Also came up with an accurate predictor of age with a TAC-seq, but they were unrelated. So dogs that look a little bit older than their actual age for the methylome don't necessarily look older for the TAC-seq. So the residuals were not correlated. I've got clock in quotes here because I think we need to be careful about just what these mean. There are many people who would argue that the deviation from this line is an actual measure of aging. I like to think about this in the way that we think about a compass. So a compass will point towards towards magnetic north, but a compass is not a magnetic field. It's a measure of a magnetic field. Similarly, this deviation is not aging. It's some measure of aging, just what I don't think we know yet. Happy to talk about that more later. Noah's group has been looking at what these epigenetic deviations do relate to. This is a very talented graduate student who's going to be finishing up this year, Bree McCoy at Arizona State, and she's found all sorts of interesting relationships between the epigenome and environmental factors, including neighborhood stability, owner age, owner income, time spent with the animals, and more. This is a slide I got from her. I've actually not seen the manuscript yet, so I can't tell you a lot of details about the underlying mechanisms. Still waiting to hear more from Bree. She's having a committee meeting soon, and I'm looking forward to learning more about this. So finally, in my own lab, we focus on the metabolome. I've been interested in the metabolome for a long time. I started working on it about 15 years ago. I was at a meeting At the time, I was still at the University of Georgia. I went to a meeting and ran into Dean Jones, who's at Emory University. And any of you who happen to have interacted with Dean Jones will know that he is a great proselytizer for the value of the metabolome. So the metabolome is made up of all the small molecules in an organism, in any organism, whether that's a bacterium, a fungus, a plant, an animal. And it makes up both the structural molecules, the amino acids, fatty acids, carbohydrates, nucleobases, and so on that go on to make up proteins and fats and DNA and RNA, as well as, so it's the structural building blocks, but also the functional building blocks like the Krebs cycle, for example. So I think there's tremendous power in looking at the metabolome as the sort of final step in this complex, somewhat linear network of domains that ultimately describe and make up the phenotype. The other thing is that the metabolome integrates both upstream genetic information and environmental information. So it really is kind of a bridge between G and E upstream and the P or phenotype downstream. The specific aqueous metabolites that we're looking at in the dog, these are all data from samples. This is lithium heparin plasma. We run it in the Northwest Metabolomics Research Center at the University of Washington that Dan Raftery leads. And just a few results. So first of all, there's a strong signature in the metabolome of weight, of age. This is work that a technician in my lab, Maria Partida Aguilar, has been doing. She's a terrific technician who does all the bench work, but she also does all her own R coding and is helping to write up the manuscript. Really terrific. Ben Harrison, a senior scientist in my lab at the University of Washington, developed a metabolome age predictor. And here you can see, does a pretty good job of predicting age. One of the things that stands out is that above a certain age, all the predictions tend to fall below the line. Now, I mentioned before that people signing up for a study of healthy aging who sign up older dogs tend to sign up relatively healthier older dogs. They don't sign up sick dogs. But this is a pattern that we also see in other studies. So, for example, here's a paper that came out some years ago in 2019, asking why it is that in epigenetic clocks, we tend to underestimate age. So we've got predicted age, actual age, and you can see at old age, all the predictions fall below this isometric line. So we're just in the process of analyzing these data. One of the things that I'm excited about is with the dog aging project data, we can start building longitudinal clocks that actually capture multiple years of data to create more accurate age predictors. And then finally, Abby Marie was a master's student, finished up her master's degree in public health genetics, was looking at both environmental and genetic predictors of the metabolome. Again, I'm really interested in the metabolome as a bridge between genes and environment upstream and downstream aging. And Abby's work suggests that we are going to be able to do this in the dog. So first of all, this is just showing the distribution of heritability for all the metabolites that we've measured. This figure doesn't show it, but if you do a permutation test and measure the heritability, permutation heritability is about 0.05 or 5%. And so almost all of our heritabilities are significantly greater than that. And you can see that our top heritabilities are extremely high for heritabilities. More than 50% of the variance in certainly in the top 10 or 12 metabolites is explained by genetic variation. So that's pretty exciting, suggesting that we might even be able to get at the underlying molecular mechanisms associated with variation in the metabolome. And then ultimately, we will be asking how these metabolites relate to downstream traits. Some very interesting environmental data. This is just from last week, not yet corrected for false discovery rates, so take it with a grain of salt. These are average minimum temperatures at the latitude and longitude site where the dog is found. Each one of these points is a dog, and you can see some significant relationships. Interestingly, we also see a positive relationship for the temperature at which the sample arrived in choline. So choline seems to be very highly sensitive to in the metabolome to temperature. Again, not yet corrected for FDR, so I don't know if it will hold up, but interestingly, there's a relationship between some metabolites and the square footage of the property on which the dog lives. This could be a measure also of urban versus rural, perhaps, or maybe time spent inside versus outside. So we'll certainly be digging down more deeply to this. And then the last thing I want to mention just in the last few minutes, and something that I'm really interested in, this was the topic of this workshop that I was at just last week, that Don DeMeo and John Quackenbush at the Brigham and Harvard put together, thinking about the role of sex and gender in precision geroscience. So as I mentioned, there are four kinds of dogs, four kinds of sexes in dogs in the veterinary world: male, female, intact, and sterilized. And it turns out that there are big effects of both. So this is work that a former graduate student, Jessica Hoffman, who's now on faculty at Augusta University and still working on the dog aging project, did. So first of all, as you can see, neutered dogs tend to live longer. Sterilized dogs live about a year longer on average, compared to intact dogs. And there's also differences between males and females. This is the veterinary medical database data, which is veterinary colleges back in the 80s and 90s. It's an older data set in the US. And this is data from the UK, from private veterinary clinics, thousands and thousands of dogs. Again, sterilized dogs live longer. But what's really interesting is that so while sterilized dogs live longer, they die of different diseases. Sterilized dogs are more likely to die of cancer and immune-mediated diseases, autoimmune diseases. Intact dogs are more likely to die of infectious disease and trauma. You may see in the popular press or social media that people will say, you shouldn't sterilize your dogs, they will get cancer. They're more likely to get cancer according to our retrospective data analysis, but they will get it at older ages. They will live longer. I am not saying you should or should not sterilize your dog. If you're wondering, I would urge you to have that conversation with your veterinarian. He or she knows your dog best along with you. So sexual differences are recapitulated in the metabolome. These are fruit fly data. And you can see in blue, we're looking at males. In orange, we're looking at females. Each point represents a different species of these different species of fruit flies of Drosophila in the genus Drosophila over 60 million years of evolution. What I want to point out is that we can very clearly distinguish females from males in terms of the metabolome, as well as older from younger. The same turns out to be true in dogs, but only partially, and it's quite striking. So if you look at this figure, this heat map, what you're looking at are metabolites that are significantly different between the sexes, but so relatively high levels in green, low levels in magenta. So you can see that for the intact dogs, we see significant differences between males and females, sometimes higher in the males, sometimes higher in the females. For the sterilized dogs, very few changes. So whether this pattern will hold up as we gather longitudinal data, I don't know, but it's quite striking that so far it looks like the great majority of the effect in dogs is gonadal rather than genetic. There are some important differences. These tend to be younger dogs, not entirely. So we need to do a very careful model to control for all these potential confounders. Ultimately, we hope to tie genetics and metabolome, microbiome, epigenome to age-related morbidity and mortality in dogs. So this is just showing some of the age-related changes of the many that we see in this population. So that the power to do this, I think, is going to be very powerful. So, and finally, this is an open science study. So all of the data that I've shown you is or will soon be publicly available. We embargo for a year. and collaborators can have immediate access if they're working closely with us through, so everybody can access the data through the Terra platform. And we have, as I mentioned, a biobank with blood, serum, plasma, PBMCs, DNA, and hair. And so finally, I think we face a huge challenge in precision medicine in that each of us is an N of one. And we can talk about just what those challenges are and maybe some of the ways that we might address them. Looking at models with genetic redundancy, whether it's complete redundancy like flies or mice, or partial redundancy like dogs, gives us greater power to test these interactions. And we can then take those ideas back to human populations for validation. So where do we go from here? Well, of course, I'm at the HNRCA. I have not talked about diet. We have board certified veterinary nutritionists on our team, and we're looking forward to starting to ask more questions about diet. Really interested in exploring this G by E mechanism. I am interested in network models. I didn't show you any of the work that we're doing, but we're very interested in how network structure changes with age. I mentioned longitudinal analysis. Ultimately, I think there's great power to take all of these molecular markers and develop better prediction, prognosis, diagnosis, treatment, and even prevention with these biomarkers. And I hope that some of you will come work with us. We're an open science study. And finally, we're a team science study. I am one of many. There are about 100 people on our team, and I want to thank them and our funding bodies, and thank you for your attention. And I do see there are some questions already. I'm happy to go
Daniel,
there. I will
thank
pass
you.
it over to you.
Yeah, thank you so very, very much for a wonderful presentation. What I'll do is I'll turn my camera off and I'll read out the questions. And I strongly encourage the audience to put your questions into the Q&A. So if you're ready, Daniel, we've got some questions already.
Right.
Eric Miller asks, does the Dog Aging Project look at mixed breeds or others dogs whose breeds may not be fully known?
Great question. We do. So we welcome all dogs of all ages, sexes, sizes, breeds, mixed breed, purebred, and geographic region, as long as it's within the 50 US states or DC. Someday we hope to expand. So almost exactly half of our dogs are purebred by owner reporting and half are mixed breed. We have 8,000 whole genome sequences so far. And interestingly, the mixed breed dogs, the owner reported mixed breed dogs are almost always mixed breed according to the genetics. Not always, but almost always. Within the purebred breeds, about 80% are really purebred, and the other 20% have between 5 and 20% of other. Some of them are those sort of designer breeds where you've got, like a Labradoodle, but many of the mixed breed dogs have some, or the purebred dogs have some of something else. Because we have 8,000 whole genome sequences using low pass coverage with imputation, we can control for the genetics of breed in a really interesting and thoughtful way using linear mixed models. That's something I'd be happy to talk about more if you're interested.
Great, thank you. Our next question is from Joel Mason. Daniel, first and foremost, we are both delighted and very excited to have you join us here at the HNRCA and look forward to our work together. My question, I assume that identical twins or triplets also exist in dogs. If so, would it be possible within the constraints of your recruitment process to recruit such pairs, thereby helping dissect out effects of the environment on the aging process?
A really interesting question. I actually don't know anything about twinning or higher in dogs. We have talked about two things. First of all, recruiting dogs from the same litter, and secondly, recruiting dogs from the same home. And these are both aspirational. One of the things I would love to do is work with both breeders in terms of purebred dogs and with shelters in terms of mixed breed dogs with pregnant females and figure out a way to recruit individual dogs into different homes that at least come from the same litter. So they would be full sibs. On the other end, we have discussed recruiting different dogs from the same home. And initially, the way we set it up was that participants could only have one dog at a time. We were concerned about people mixing up the dogs and answering for the wrong dog. And it just makes things simpler. What people can do is if their first dog is in the project and then dies or moves and is no longer in the project, they can then recruit another one. So we don't have contemporaneous dogs from the same environment, but we do now allow dogs over time from the same environment. I would love to work on ways to do both what you've talked about, or at least within the same letter, and simultaneous dogs within the same home. If I can convince my IT team to get on board, it makes things very complicated. But for the reasons that you raise, it's a really interesting question to ask.
Thank you. The next question is from Christopher Wiley. Really enjoyed this, Daniel. Perhaps I missed it, but you discussed IGF-1 as a master regulator of dog size and how it got you into dog aging. But is it also a master regulator of dog aging?
We still don't know. It's a great question, Chris. It's what got me into aging, and I tried to answer it using breed average data, and it just wasn't, we didn't have enough power. We aren't currently measuring titers of IGF-1, although I think that would be interesting. So we just don't yet know. I will mention there's a for-profit company that was created by Celine Haliwa called Loyal, and they are developing drugs that target IGF-1. Their hypothesis is that at least large breed dogs, and maybe all dogs, would be longer lived if we knock down IGF-1 once they're adult. And so they have a drug that I think it's proprietary, and I don't know anything about this, but my understanding is that this proprietary drug for large breed dogs will knock down IGF-1. They're doing a clinical trial, and maybe 4 or 5 years from now, we'll know if knocking down IGF-1 in large breed dogs increases their lifespan. I will be very interested to see their results.
Great, thank you. The next question is from Jiangdong Wang. Great project and talk, thank you. Saliva metabolites are suggested to reflect the health status of an individual in humans. As a non-invasive sample for metabolomics, I was wondering if dog saliva could be investigated in the dog aging project and could offer a new alternative material for research to reveal molecular mechanisms of aging, example comparison of human and dog saliva composition.
Thank you for that question, especially because it reminds me to tell you one thing that I forgot to mention, which is that for years I've been really excited about trying to create another dog project, because one doesn't seem to be enough, that would be a matched cohort of humans enrolled in a human long-term longitudinal study with their dogs enrolled in a new long-term longitudinal study that mirrors the dog aging project. So that would get partly at your question. And the answer is, I don't know, but I'd love to talk with you more about this. For the metabolomics that we do, we need about 50 microliters, which isn't that much. I'm guessing that we could get that much of material off a cheek swab. The cheek swabs that we currently collect are designed just for DNA collection. It would be interesting to think about doing something like that in a population of dogs at the clinic, rather than, so we send the cheek swabs to people's homes because DNA is stable for a long time. Obviously, the metabolites are not. So the challenge would be to figure out how to get the metabolites into the freezer quickly to preserve them. So I think doable, and certainly I'd be interested in, if it is doable, maybe exploring writing an ancillary study grant to fund something like that. So I'd be happy to talk with you offline about that.
Great. Daniel, we have 2 more questions and we're coming up to time. So the first one is from Lynn Hughes. Dr. Promislow, can you speculate why the metabolome variation is so high in intact dogs and not so much in sterilized dogs?
Thanks for that question. Um, so what it suggests to me, and I can only speculate, is that there is a very strong effect of gonadal hormones on dogs. And in the sterilized dogs, they're lacking major sources of testosterone, estrogen, estradiol, and so on. And so we simply don't see the impact of those hormones. Obviously, there are still hormonal sex differences after gonadectomy between males and females, but I can only guess that they're much subtler in terms of their effect on the metabolome.
Great, thank you. And last question is from Cy Das. Great talk. Do any of the surveys capture the lifestyle behavior of owners? If yes, would within breed differences based on owners be examined?
Such an important question. So thus far, just to make our lives easy or easier, we filed our proposal, the initial proposal and the one that we're going to be submitting from Tufts. Thank you, Megan and Chris and the Tufts team for all your work. We have an IRB exemption. So we don't ask people about we don't ask them any questions that would require IRB approval. We hope to change that. So we don't know if someone smokes. We do know if their dog is exposed to smoke. We have some basic questions about demography, age, income, education level, and what we know about the neighborhood that they live in, but not their sort of health behaviors, diet, lifestyle, and so on. So that's something that I'm very interested in pursuing once we have the next cycle funded and we can begin to put the time into designing some IRB, some questionnaires that would require IRB approval.
Great. Well, those are all the questions. Thank you so very, very much, Daniel, for a wonderful seminar. And thank you to the audience for being so engaged and asking such great questions. That concludes our seminar.
Thanks, everyone.